Hello guys, well done for logging on and finding the lesson. For those of you that don't know me, my name's Mr. Davis. I'm one of the science teachers at the St Leonard's Academy, and I'm going to be recording a quick lesson for you guys about animal and plant cells and the techniques that we use to look at them, i.e. microscopes. So the lesson today is going to be covering how microscopes work, how we can calculate things like magnification, and looking at the different organelles that you find in animal and plant cells. We're going to do this because, firstly, microscopes are an incredibly useful scientific tool that we use within the scientific community. And by understanding the different organelles that you find in animal and plant cells, we get a better understanding of how those organisms can function. Now, for this lesson, guys, you are going to need a pen or a pencil and a notebook or some pieces of paper. So if you haven't got those to hand now, what I suggest you do is quickly pause the video, run off, go grab yourself a pen and a pencil and a notebook or a piece of paper, and then get yourself settled back down in a nice quiet space so you can get on with your learning. So pause the video now, go off and do that, and then head on to get yourself set up and ready to learn. Okay, well done, guys. For those of you, well done. Yeah, well done for getting all your stuff together and being ready to learn. So the first part of the lesson, we'll be looking at magnification and resolution. These are two aspects of microscopes that you need to understand about. We can compare the two main types of microscopes that we use within the scientific community. We're going to work out how we can calculate magnification. We're going to look at the different organelles you find in both animal and plant cells, and then we'll end on a nice 6-mark question to give you a good chance to demonstrate your understanding of the animal organelles of an animal and plant cell. So, moving on quickly and promptly. What I'm going to get you to do now is a very quick do it now, guys. This is just a quick recall from years 7 and 8. What I'd like you to do is pause the video, give yourself a couple of minutes, and just see if you can write down the names for each of the parts of the microscope that you can see on the screen in front of you. So pause the video now, have a quick go at that, and we'll go through the answers in a few moments. Okay, guys, well done for having a go. I don't know how many you got right, but we're going to go through the answers now. If you haven't got the correct answer, just jot down the correct answer, correct your answer, and if you've got any wrong, also correct them and give yourself a score out of 7 that you got right. So initially, part A of the microscope is known as the eyepiece or ocular lens. This is the bit we put our eyes up to and look down. Part B is the arm. Can also be referred to as the bridge, but more commonly known as the arm. C is the focus adjustment. D is the objective lens. E is the stage. That's where we mount the sample that we're going to be reviewing. F is the light source, and G is the base. So give yourself a mark out of 7 for those you got correct, guys. And then also, as I say, correct any answers you got incorrect. Okay, guys, so magnification and resolution. These are two really important factors of microscopes. The magnification is the degree to which the size of an image is changed, how big we can make something look when we put it under the microscope. Now, the detail that we obtain from a microscope when we're looking down at that image is referred to as its resolution. Now, the definition of resolution is it's the smallest distance between 2 points that can still be identified as 2 separate points. So for those of you that have not been taught by me before, what I try to do is for the key things that I need you to have in your notes, I generally surround in a red box. So as you can see, we've got 2 red boxes on the screen now, guys. And what I would like you to do is to just quickly pause the video and just copy those 2 definitions down into your books, the one for magnification and the one for resolution. So pause the video, copy those down into your books. And once you've done that, press play again, and we'll run on with the rest of the lesson. Okay, well done for having a go at that, guys. Now, we're going to move on. So we're going to now look at calculating magnification. Now, magnification can be calculated effectively in 2 ways. If you're using a light microscope, you can calculate the magnification by multiplying how much the magnification of the objective lens and the eyepiece lens. So the total magnification is equal to the objective lens times the eyepiece lens. If I was to give you an example, if you say had an objective lens of times 10 and you had an eyepiece lens also of times 10, the total magnification that you would expect when you look down the microscope would be 100 times. That's 10 times 10, giving you 100, and it's 100 times magnified. Now, again, you can see I've jotted a red box around that equation. It's really key that you're able to recall that equation, guys. We've got another equation as well, which is the other way in which you can work out your the level of magnification that you're producing with a microscope, and that's by looking at the image size that you're generating with your microscope, and also the actual size of the object you're looking at. Now, I've put an equation triangle next to the image size calculator equation, and that label means that you can work out any facet of those three things with the other two. If you've got the other two bits of data, you can work out the third of them. Now, if you haven't used formula triangles before, and I'm pretty sure most of you will have come across them, you can see if the two, you can cover up one of the corners, and that will relate to you what you have to do mathematically to the other two bits of information you've got to be able to calculate that corner. Now, as you can see, we've got image size equals actual size times magnification. What I would like you to do, guys, is pause the video very quickly, copy those two equations down into your book. Also copy the I am triangle, the image equals actual size times magnification triangle. And I'd also like you to try and draw your own triangle for the total magnification equals objective lens times eyepiece lens. So pause the video now, copy down those two equations and draw down those two triangles, and we'll go through them, make sure you've got it all right before we answer that last question. Okay, guys, well done for having a go at that. So if you did have a go at trying to draw that triangle, it should look something like that, with total magnification being at the top. That's what TM stands for, and objective lens being in one lower corner and eyepiece lens being in the other lower corner. Now, if you're trying to calculate using a formula triangle, as I say, you just cover up the corner that you're trying to find out, and then you either multiply or divide according to what position on the triangle the two remaining objects that you know are. Now, we've got a question at the bottom of this slide, and it says that if a microscope has an objective lens of 5 times and it increased the size of an image of a 3mm object to 3 centimeters, how powerful is the eyepiece lens? What I'd like you to do, guys, is to pause your video, have a go at this question, and then we'll go through the answers after you've had a go. I will give you a bit of a heads up, guys. To do it properly, you are going to need to be able to use both of the triangles to be able to calculate the answer to that question. So pause the video now, have a go at that question, and then we'll go back afterwards and have a go at the answer afterwards. Okay, well done for having a go, guys. So what we're going to do is we're going to look at this question. If a microscope with an objective lens of ×5 increased the size of an image of a 3mm object of 3 centimeters, how powerful is the eyepiece lens? Well, we've got a lot of information there, guys, but we need to begin with, because the only one we've got with the information we've got the image size and the actual size of the object, and that's going to give us our magnification. So if we do that first, and that will be image size, we're covering up the magnification in the bottom corner of that triangle. Image size divided by actual size will give us the level of total magnification, or magnification that the object is undergone, and that is 30 divided by 3 equals 10. Now we have to do 30 divided by 3 because we've standardized the units. What we've done there is 3 centimeters is equal to 30 millimeters. So I've just converted that before I've added the number into the equation, and that gives us a total magnification of times 10. Now we now know the total magnification, and we know the objective lens power, which is a times 5, and that means that we can quite easily work out how powerful the eyepiece lens is. So again, we would cover up that E on our triangle, and we know that it is, as a result of covering that E, total magnification divided by the objective lens will give us the eyepiece lens. So if we were to follow that equation out and input all the numbers, we would see that 10, which is our total magnification we calculated on our last sum, divided by 5, which is the level of magnification that we get in the question of the objective lens, will provide us with 2 times as the level of magnification that the eyepiece lens has. So that's the 2 triangles used in conjunction to enable us to work out that question. Okay, well done for having a go at that, guys. It is quite a tricky question because because you do have to use the 2 triangles to be able to work the question out properly. Okay, light microscopes. These are awesome things, guys. Basically, light microscopes work by having 2 curved lenses at either end of a cylindrical tube, and they allow us to magnify quite greatly something on the underneath of the lenses as we look through them. Now, it was first invented by a guy called Robert Hooke, and he discovered he invented his microscope and discovered cells at the first time. So before we had microscopes, we had no idea that we could look at these small things in such detail. And so he was the first person to discover cells, and he used this microscope that you can see on the board. This is a model of it. It's not the actual microscope, but this is a model of the microscope he used. And with that microscope, he was able to magnify objects up to 30 times, which is quite impressive when you consider this was in 1665. You know, this is the year of the bubonic plague. This is the year before the Great Fire of London. So this is a long time ago, guys. Now, or shortly after that, or in the prevailing years after that, by improving the quality of the lenses that went into this designer microscope, we eventually was able to magnify things up to about 270 times, which is a really vast improvement over 30 times. However, modern microscopes, the types that we have at the academy, and that are quite commonly and frequently found in science labs, they're now able to magnify objects up to 1,000 times. And that's getting close to the limit on which we can magnify objects using a light microscope. And that is all being driven by the improved quality of the lens. Having no imperfections in the lens means that the light travels through the lens as it should do, with no bouncing off, no interference, and we're able to get very clear and precise images. So electron microscopes, these guys are amazing. They were invented in the 1930s. They're absolutely massive. You can see, this is actually, the image you can see in front of you here is actually quite a small electron microscope, and they can be almost the size of a room in some instances. And these work by using a beam of electrons to build up the image. And they allow you to magnify things to a much, much greater level. Up to 2 million times you can magnify with an electron microscope. These allow you to see even down to viruses in quite good detail. And it does allow us to look into all the individual bits that you can see in the inside, the different organelles you can see inside a cell. Now, the reason we're not all using electron microscopes, there's a couple of reasons really, guys. Firstly, light microscopes are relatively small and portable. I've got one here, in fact, that I can just pick up and wave around in front of you. This is one I use with my son when we're looking at different things we find in the garden. They're small, they're portable, they're also relatively cheap. So you can buy a light microscope for a fairly decent one for between £400 to £500, which is relatively cheap when you look at buying even a cheap electron microscope, you're not going to have a lot of change out of £10,000 to £20,000, and that's still a relatively cheap one. You can spend, you know, quite a bit more than that on a really high-powered, high-quality electron microscope, and that's one of the reasons we don't have one at the school. Now, the reason that electron microscopes give us this much, much better image is all to do with the wavelength of an electron. Now, I'm not going to go into too much detail of the duality between particle and wave theory. That's a level of quantum mechanics that you wouldn't really need to learn unless you decided to do a PhD in the subject. But electrons do have a wavelength, and it is absolutely tiny. Now, visible light, when we use visible light, and the wavelengths of light that are going into our eyes ranges between 400 and 700. That's a rough approximation. Whereas the wavelength of an electron, sorry, it's 400 to 700 nanometers. I'll give you a reference, an actual unit. The number is useless in science if it doesn't have a unit. So 400 to 700 nanometers is the wavelength of visible light. An electron has a wavelength of 1 nanometer, and it's that much smaller wavelength that allows us to get much greater magnification and also much greater resolution. Now, you'll always be able to tell, well, you can generally tell when you're looking at an image that has been produced by an electron microscope, because all electron microscope images are black and white. Light microscopes, you get the full range of the visible light spectrum, and you can create images in any light, any color that our eyes are able to detect. Whereas an electron microscope can only produce images in black and white. So they only produce black and white images. They're massive and very expensive. Light microscopes, however, smaller, more portable, much cheaper in comparison to an electron microscope, and you can produce images in color. Okay, guys, so a very quick question covering some of the stuff that we've been talking about now. Just wanted to have a very quick few moments on this. What happens to an image when I increase the magnification of the objective lens and decrease the eyepiece lens by the same amount? I'm not going to get you to pause the video, guys. Just very, very quickly in your heads, read those three statements and then have a guess at which one you think is the correct one. A, B, or C. So take a few moments to read those now. Okay, guys, if you selected B, then you are indeed correct. The image would stay the same as the magnification. The image would stay the same as the magnification is the combination of both lenses. Remember, we were talking about that triangle, and total magnification is the objective lens multiplied by the eyepiece lens. So it's a combination of both pieces, and that's why B is the correct answer. So a very, very quick exam style question. This is actually lifted straight out of an exam. These are the kind of questions you might find in your GCSE. And it's just simply named 2 advantages of using an electron microscope to examine cells over a light microscope. So I'd like you to pause the video here. It's a 2-mark question, so give yourself no more than 2 minutes to answer this question, and just have a go at seeing if you can come up with a couple of reasons as to why an electron microscope is advantageous to use over a light microscope. Let's pause the video now and have a quick go at the question, guys. Okay, well done for having a go at that, guys. So if we were marking an exam, we would have this as our mark scheme. This is what we would be allowed to give correct marks for. So give yourself a mark, at 1 or 2 marks. If you put any of those any two of those answers, either the image would be clearer, or the image has more detail, greater resolution, or the image has a larger image or greater magnification. Any of those facts would be considered as a correct answer to that question. Well done for having a go at that, and well done for and if you haven't got it right, not to worry, make sure you've jotted down in your notes the correct answers, and we're going to move on now to the next part of the lesson. And that's all about the animal and plant cells, guys. So what I'm going to get you to do is we're going to break this down a little bit. What I'd like you to do is, on a fresh page, if you can, in the middle of your page, I'd like you to draw this animal cell that you can see in the middle of the screen into your page. I would then like you make it quite large, maybe 10 lines. If you're using A4 paper, maybe 10-ish lines in the middle of your page. And then what I'd like you to do is label all the different organelles that are in this diagram. Now, again, this is a bit of a recap. Some of these organelles you will have been taught about in year 7 and 8. We've got a new one that's added into this one that you may not have come across before, but try and label as many as you can. And also, I'd like you, when you're labeling them, to try and include the functions for each of the organelles you label. Pause the video, give yourself a good 10 minutes to do this, guys. Don't worry, or however long it takes you to do. We will be going through the answers when you come back to the video. So just pause the video now, draw the cell out, label it up, and then write the functions for each of the organelles that we've got on that diagram. Okay, well done for having a go at that, guys. So hopefully now you've got your animal cell drawn in the middle of your page. You've been able to label all of the different organelles we've got there. As I say, there is a new organelle that if you weren't able to label, I fully understand, and we'll cover that in a bit more detail when we get to it. So the first of the organelles we've got is the nucleus. And then after the nucleus, we've got the cell membrane. Then after the cell membrane, we've got the mitochondria. Then we have the cytoplasm. And this little dot is the new one that you may not have come across. This is known as a ribosome. Now, I will be covering the function of the ribosomes in a bit more detail in a little bit, but they're basically the site of protein synthesis. Okay, so the nucleus. The nucleus is the nucleus contains the DNA of the cell, and it controls all the activity of the cell or in the cell. So that's the function hopefully you put alongside your label of the nucleus. And we're going to move on now to the cell membrane. The cell membrane is there to control what enters and leaves the cell. It's a bit like the gatekeeper of the cell, so it can control what goes in and what comes out of the cell. Moving around to the mitochondria. The mitochondria is the site of cellular respiration. It is where energy is released from the food that we eat. The glucose that we eat is there combined with oxygen, and it releases energy that our cells can use. The cytoplasm. Now, this is a jelly-like substance that actually is basically where all the chemical reactions of the cell can take place. And finally, the ribosomes. As I said earlier, it's the site of protein synthesis. And this is basically how the nucleus is able to control the cell. So the nucleus has DNA in it. That DNA can send signals to the ribosomes and get them to produce different proteins. And proteins are really, really useful. They form they govern so much of what the cell does, from enzymes to different markers and cell gates and things that allow stuff in and out the cell. There's all sorts of things that are controlled by the proteins of our body, and the ribosomes are how those proteins are made. Okay, guys, so hopefully you've got a bit of an understanding now of all the different organelles that you need to know at this stage of your education that can be found inside an animal cell. Okay, so we're moving on. We're going to move on now to look at a plant cell. Now, what I'd like you to do is, again, if you can, if you've got enough pieces of paper or a big enough notebook, flip to a fresh page, give yourself a title at the top of a plant cell, and in the middle of that fresh page, I'd like you to have a go at drawing a diagram of the cell that you can see in the middle of the screen. And again, we're going to be going through that same process, guys. What I'd like you to do is label all the different organelles, and they're all there to be labeled, but I'd also like you to include the functions for each of the plant-specific organelles. So you will, if you the quick quick-witted of you will have noticed that there are more labels on this diagram. And that's because there are a number of organelles that you will find in a plant cell that you will not find in an animal cell. All the ones you find in an animal cell, you can find in a plant cell, and they are included on this diagram. But there are 3 organelles on this diagram that aren't found in animal cells. And for those organelles, guys, I'd like you to have a go at labeling the, including the functions for those organelles as well into your labels. So again, guys, we're going to give yourself a good crack of time here, a good 5-10 minutes, however long you need. Draw out this diagram in the middle of a fresh page, big diagram, make it, as I say, about 10 or so lines, and nice clear labels. And as I say, where possible, if you can remember the function of the organelles, add them in. But on this diagram, you will only need to add the functions for the organelles that you that are specific to plant cells. So pause the video now, give yourself a good 5-10 minutes to draw this diagram and then label it and add the functions of the plant specific organelles. Okay, guys, well done for having a go at that. What we're going to do is we are going to now very quickly go through the answers to this question. So I'll go through the labels of this diagram. So again, we're going to start with the nucleus. We then come down to our mitochondria. I've coloured these the same colour as on the previous diagram. They're not pink. They don't look pink when you look at them down a microscope, but I've just done that to enable them to be a bit more clearly picked out of the from the diagram that you're drawing. Then we have cytoplasm, and again ribosome, and finally cell membrane. Now you will notice, guys, that all 5 of those organelles that I've labelled up here all can be found in animal and plant cells. These are the cells, these are the organelles that both types of cell have in them. And both types of cell rely on very heavily to have them within them. So the 3 organelles that we haven't covered yet are the cell wall, the vacuole, and the chloroplast. Now, I've coloured these chloroplasts green because that is literally the colour they are. Chloroplasts are the reason that plants, that the leaves of a tree are green, effectively. And it's because, again, something you don't really need to know, but it's basically because plants can use all the different colours within the visible light spectrum of light except green. So they can use yellow light, red light, blue light, orange light, purple light. The only light they can't use is green, and that bounces back. All the other lights are absorbed. Green light is reflected and it's bounced back into our eyes, and that's why the leaves actually look green in the first place. But it's also because these chloroplasts are in very high numbers in some of the cells inside the photosynthetic parts of the plant. So we're now going to go through the different functions of the plant-specific organelles. The first of these is going to be our cell wall. Now, the cell wall is it's made out of cellulose. I haven't included that on this slide, but I would advise adding that into your function. But the cell wall is present in a plant cell to both protect it and to give it structure. And it's very important for certain plants that cell wall has a very strong structure. It is, for example, why grass is able to grow quite tall without having that woody texture that some of the trees and bushes and larger plants are able to achieve. They use a combination of the vacuole and that cell wall to enable them to have that upright structure. So the function of the cell wall is to provide the plant cell with protection and structure. Now, the vacuole, this is a part inside the cell that can store cell sap. Now, that's basically quite a sugary solution. It's got lots of nutrients in it, and it can be a reserve that the cell uses for storing its glucose in, for example, for respiration. But it also combines with the cell wall to enable the the plant cell to remain structurally sound, and it does that by absorbing fluid and pumping fluid out when it needs to. It's a complicated process, and it isn't something that you guys need to understand at this point of your education. Okay, so that's the function of the vacuole. As I say, it's to store cell sap and to give the cell its structure, or to assist the cell wall in giving the cell its structure. We finally move on to chloroplasts. Now, chloroplasts are really what makes plants plants. Plants are able to photosynthesize. That means they're able to produce their own food from sunlight and carbon dioxide and water. They combine those three factors together and they produce a sugar that their mitochondria are able to respire with. So they contain chlorophyll, and they are effectively the site of photosynthesis. But that in itself is a whole series of lessons that we'll get into at another point in time, guys. Just at this point in time, as long as you know that the chloroplasts contain chlorophyll and they are the site of photosynthesis, that's all you need to know at this stage. Okay, guys, so we've covered all the organelles of a plant cell, all the organelles of an animal cell. And we're going to move on to our exit ticket. The last bit of work I'm going to get you guys to do. I've been a bit mean in that I'm asking you to do a 6-mark question. These are really important, guys, because they can actually relate to a very large percentage of your overall grade when you're sitting an exam. If you've got a question that's, if you do an exam paper that's maybe 35 marks and you've got a 6-mark question at the end of it, now, even if you only do 3.5 worth of marks in that 6-mark question, that's still 10% of your overall grade. So these 6-mark questions, I'm really going to push them with you guys. You need to be able to answer these confidently. Now, when you get a 6-mark question, there's a lot of things you can do that will help you answer the question. The amount of times I mark exams and people have either not read the question properly or not understood the question properly, or their answer doesn't relate to the question, it's really infuriating. So one technique you can use to make sure you do a really good answer to this style of question is bug the question. Now, what that basically means is you box the command words, you underline the key terms, and you constantly go back over the question, making sure that what you're writing is an answer to that question. So an example of that would be, in this instance, we would box the words compare and contrast, because they're the command words of this question. They're telling us what we have to do in the question. And then underlying the key terms, well, organelle and animal and plant cell. They're the 2 key terms that we need to be able to answer the question. And then constantly, whilst answering that question, I would like you to go back to the answer and go back to the question, reviewing what your review the question and check that what you're writing relates to it. Now, if you guys are in any of the triple science lessons, what I would like you to do, guys, is pause the video now and have a go at this question. Once you've had a go at this question, we're going to go through the answers. I've got a model answer set up for you, and we'll go through the mark scheme so you can have a go at marking your effort yourself. So that's for the triple science students. Pause the video now, have a go at the question, and then once you've had a go at the question, we can go over the answer. For the rest of you, I've just given the triple science guys a chance to pause the video. For the rest of you guys, what I'm going to get you to do is I'm going to also give you a bit of a highlight. Now, in an exam, you won't get this. This is not part of the exam material that will be provided to you, but I've included the success criteria for this question because I am aware that for some of you guys, 6-mark questions are terrifying. And so we're going to walk you through a little bit of what you need to do. So compare and contrast means you're looking for the similarities and the differences in the organelles between animal and plant cells. That's what this question's asking you. Similarities and differences in the organelles between animal and plant cells. So I would say what you would need to do to ensure you're getting the right mark is look firstly for the similarities. So we're looking at naming the organelles that you find in both animal and plant cells. There's a big tick for your similarity line. Then you want to look at the organelles only found in plant cells, and that gives you your contrast, your differences that you've got. And then, because you're looking at these organelles in a bit of detail, it would be prudent for you to explain the role or the function of at least two of the organelles that you find in plant and animal cells. And I would say that you would also need to, that's just, again, you're looking at you're comparing your similarities, and then also do your explaining the role of an organelle found only in a plant cell. And that's your contrast, guys. So there you've found the success criteria there will give you all that you need to be able to answer the question. Just for those of you that do need a little bit of extra support, we've got some sentence starters here just to get you underway with your writing. Sometimes those mental blocks can be hideous and you can't get the words out. You know what you want to say, you can't get the words out. These sentence starters are a really good way of just getting the words to start, and then hopefully your ideas will be able to flow onto the page a little bit better. So pause the video now, guys. Have a go at this question. Once you've had a go at this question, we'll go through the answer. I'll go through the mark scheme so you can have a go at marking your work, and then I'll provide you with a model answer that I've written as to how I would answer this question if I was in an exam. So pause the video now, guys. Normally give yourselves maybe 6 minutes, but seeing as this is maybe the first time you've done a 6-mark question, or certainly you may not have done a 6-mark question recently, give yourself a little bit longer if you need to. Take as long as you need, really, guys. In an exam, you'd be looking to spend nowhere more than between 6 to 10 minutes on a 6-mark question. But in this instance, because it's the end of the lesson, take as long as you feel you need to make sure you've got a real good understanding of the comparisons and contrasting aspects of the organelles found in animal and plant cells. So just pause the video, give yourself 10 minutes or so to answer this question, and when you come back on, we'll go through the answer. Okay, well done, guys. We're nearly there. We're just going to very quickly. Here's the mark scheme. So if I was marking the exam and that was one of the questions that would marked, this would be what I would be looking to find in your answer to be able to give you marks. So if you have listed at least 4 different organelles that are present in both animal and plant cells, then you can give yourself a mark, okay? We want to be having more than just one. You know, if I was in a really good mood and you were really kind to me and behaved well in my lessons, I might consider giving you a mark for 3 different organelles. But, you know, for the most part, guys, if you've got 4 different organelles, you can give yourself a mark. Now, if you've also listed 2 organelles that are only present in plant cells, there's another mark there. So we've got our comparison, we've got our contrast of what organelles are found in plants. And then if you describe the function of at least 2 of the organelles that you've labelled that are present in both animal and plant cells, again, we're getting that comparing, that similarity. And then another mark can be given if you've described the function of an organelle that's only present in plant cells. Again, there's that contrast, that difference between the 2. And also, as a full in-depth look into the organelles of plant and animal cells, if you've explained why that organelle is only found in plant cells, then you're fully answering that question. And also, you do have to, during your exams, wherever possible, be writing in full sentences with a clear structure. Quite often there's a cap, even if you write down every fact that you know about this subject, if you haven't presented it in a way that's clear and easy to read with a clear structure, there is a cap on the level of points that we're able to give you, which is why I've got it down there as the 6th mark. So if you've done the 5 marks and you've got those 5 points perfectly in your answer, that's brilliant. If you've written it in full sentences with a clear structure, absolutely amazing. Pause the video, guys, use this mark scheme, give yourself a mark out of 6, and then I'm going to show you, once you come back on, I'll show you my model answer and how I've awarded myself the 6 marks. Right, well done, guys. Hopefully you've done well on that question. I'm now going to throw up a model answer. If you want to, you can pause now and have a go at redrafting your answer to give yourself a better mark. But if not, here's my answer. Oh dear, I didn't realise that was going to be in the way. Let's see if I can move this out of the way. Can I? I can. I've gone down the bottom of the screen. Right, okay, we're down here at the bottom of the screen now. Okay, so the mark scheme that we've got in front of you, so you can see where I said the organelles that are present in both animal and plant cells include the nucleus, the cell membrane, the cytoplasm, the mitochondria, and the ribosomes. So I've got this color coded in this dark blue, and that's definitely a tick over here. I've definitely listed 4 of the different organelles found in both animal and plant cells. I think I actually listed 5 on that occasion. And then you have listed at least 2 organelles present in only plant cells. Well, I've included that by saying plant cells have extra organelles not found in animal cells. These include the cell wall, vacuoles, and chloroplasts. So I've labelled the organelles that are present in the animal and plant cells and the organelles present only in plant cells. So I've done my similarity and my differences, my compare and my contrast. Now I've described the function of a couple of the different organelles that we have in both animal and plant cell. I've stated that mitochondria release energy stored in glucose and ribosomes are the site of protein synthesis. These organelles perform the same function in both types of cell. So there's me saying, basically, I've including that description of the function of 2 organelles that are present in both types of cell. I've then included that chloroplasts are the sites of photosynthesis. That is the function of an organelle that is only present in plant cells. And I've also said that they are only found in plant cells because animal cells do not need to photosynthesize. So by including that line at the end, then that gives me my 5th mark. I would argue that this is probably fairly well structured as an answer, and I would want to be giving myself a 6th mark for this if it's laid out in this fashion. So guys, hopefully you've been able to get a good understanding now of the different organelles found in animal and plant cells. You've also got a good understanding of the 2 different types of microscopes that we can use to view those cells. There is work that's been set for you guys both on Teams and on Seneca. If you wanted to expand on your learning, by all means, go ahead and have a go at those exercises. Other than that, well done for logging on. Well done for sticking with me this long. It's been a bit of a whistle-stop tour of animal and plant cells and microscopes, but hopefully you've been able to learn something. And I look forward to the next lesson I'll be delivering from this room. Take care, guys. Have a lovely week, and I will see you soon. Take care. All the best. All right, I need to...